This protocol aims to guide the fabrication of self-assembled three-dimensional bundles of longitudinally aligned astrocytic somata and processes within a micron-sized hydrogel microcolumn for use as an in vitro neurobiological model or as a neuroregenerative therapy.
Trauma and neurodegenerative disease are particularly detrimental to patients due to the restricted neurogenetic capacity and inhibitory environment that limit regeneration in the central nervous system.
Engineered astrocytic bundles address the limitations of current treatment strategies by attempting to replicate key neuroanatomical features and glia-mediated developmental mechanisms to promote targeted cell migration and axon path binding.
The architecture of these astrocytic scaffolds is primarily inspired by the aligned astrocytic processes in the glial tubes of the rostral migratory stream.
Begin by preparing a 3% weight per volume agarose solution in DPBS in a sterile beaker. Maintain the heating and stirring to prevent the solution from solidifying prematurely. Insert an acupuncture needle into the bottom opening of a bulb dispenser. Place a capillary tube over the exposed needle and secure the tube by entering part of it into the rubber section of the bulb dispenser cylinder. Cap the cylinder to complete the assembly. Transfer 1 milliliter of liquid agarose to the surface of an empty petri dish. While pinching the rubber cap of the bulb dispenser, insert one end of the capillary tube into the agarose and release the pressure on the cap to draw agarose into the tube. Place the bulb, tube, needle,